Imagine trying to find one specific grain of sand on every beach in California. That's what designing DNA used to be like. Until 3 weeks ago, when scientists at Rice University did something that sounds like science fiction. They taught artificial intelligence to design DNA circuits, and it worked better than anyone expected. Here's the thing most people don't realize. We've been able to program living cells for years. Scientists can make bacteria produce insulin, engineer immune cells to fight cancer, even create organisms that eat plastic. But there's always been one massive, frustrating bottleneck. Finding the right DNA sequence is like being handed a Rubik's Cube with 1 billion sides and being told, solve it, but you only get one guess per month. The math is brutal. A single genetic circuit might have millions of possible designs, and testing each one the old way, that could take decades. Enter Classic. No, it's not a new streaming service. It stands for Combining Long and Short-Range Sequencing to Investigate Genetic Complexity. And yes, scientists are terrible at naming things, but stick with me, because what this does is genuinely mind-blowing. Rice University's team, led by bioengineering professor Caleb Basher, figured out how to test not hundreds, not thousands, but millions of DNA designs simultaneously. Let me put that in perspective. What used to take a research team years to accomplish now happens in a single experiment. Here's the clever part. They combined two types of DNA sequencing that scientists usually use separately. Think of it like using both a telescope and a microscope at the same time. Sounds weird, but it gives you information neither tool could provide alone. Long-read sequencing captures the full picture of each DNA circuit, thousands of genetic letters at once. Short-read sequencing is faster and more precise over short distances. Most researchers pick one or the other. The Rice team said, why not both? And that decision unlocked everything. They built massive libraries of genetic circuits, each one slightly different, and inserted them into human kidney cells. Then they added a brilliant tracking system. Each circuit got its own unique barcode, like a genetic QR code. When the cells activated certain genes, they would glow. Brighter glow meant stronger activity. Using short-read sequencing, they could identify which barcode, which DNA design, was in which group of cells. Suddenly, they had a massive dataset connecting DNA sequences to actual cellular behavior, hundreds of thousands of data points. And that's where the AI comes in. Machine learning models are hungry for data. Feed them enough examples, and they start recognizing patterns humans never could. The Rice team trained their AI on this enormous dataset, teaching it to predict which DNA designs would produce specific cellular behaviors, even for sequences they'd never physically tested. The validation results? 40 predictions tested manually, 40 perfect matches, 100% accuracy. Co-first author Katij Rai put it bluntly, this was the first time AI could be used to analyze circuits and make accurate predictions for untested ones, because up to this point, nobody could build libraries as large as ours. Now you might be thinking, okay, cool science experiment, but why should I care? Fair question. Here's why this matters to you, even if you never set foot in a biology lab. Cancer immunotherapy, those treatments where doctors reprogram your immune cells to hunt tumors, relies on genetic circuits. The faster we can design better circuits, the faster we get better treatments. Same goes for diabetes therapies, genetic diseases, even aging research. Beyond medicine, we're talking about industrial applications, bacteria engineered to produce biofuels, clean up oil spills, manufacture materials without toxic chemicals. Every one of those applications needs precisely designed genetic circuits. Classic just made that process exponentially faster and cheaper. Scientists recently stumbled onto something completely unexpected. It turns out there's almost never just one perfect DNA design. Instead, they discovered that the genetic circuits they're trying to build often have multiple solutions that work equally well. Imagine a lock that doesn't need one specific single key. It'll actually accept several different keys, as long as they all share a few common features. This fundamentally changes how we approach genetic engineering.
We can stop obsessing over finding that one single optimal sequence. Now, engineers can choose from a whole menu of working designs, picking one based on other factors like its cost, its stability, or just how easy it is to manufacture. And what's even more intriguing is that this redundancy, it perfectly mirrors how natural evolution works. Nature doesn't optimize for perfection, it optimizes for good enough, with backup options. That's the secret sauce for how life has survived billions of years of environmental chaos. In practical terms, this means if a new genetic therapy works beautifully in the lab but fails when you try to scale it for mass production, you don't have to go back to square one. Engineers can simply switch to an equivalent design that's easier to manufacture. It is the difference between having one fragile path to success versus having multiple resilient pathways. And that is exactly what you want when you are engineering something as complex and unpredictable as a living cell. This flexibility is fantastic news. It means biological systems can be more robust, more adaptable. If one design fails under certain conditions, there are backup options ready to go. But the bigger picture here, what Rice University really demonstrated, isn't just a better way to design DNA. It's a proof of concept for how AI and biology can work together as true partners. We're watching the emergence of a brand new field where machine learning doesn't just analyze biological data, it actively participates in biological design. The economic implications are staggering. The global synthetic biology market is projected to hit $30 billion by 2030, and technologies like this could accelerate that timeline by years while slashing R&D costs by up to 70%. We are talking about democratizing genetic engineering. What once required a multi-million-dollar lab and a decade of work might soon be achievable by a small biotech startup in a matter of months. Think about how AI transformed drug discovery, protein folding, medical imaging. Now, apply that exact same revolutionary potential to genetic engineering. That is the door that was just opened. And this isn't hype. The research was published in Nature, the journal where only the most rigorously validated, groundbreaking work appears. This is peer-reviewed science that other labs will now build upon. So where does this go next? The Rice team is already working on more complex circuits, testing the absolute limits of their AI predictions. Other research groups will adopt and improve this technique. Within 5 years, we will likely see therapies designed using this approach entering clinical trials. But here's the really ambitious part. Researchers are now talking about genetic circuit compilers, software where doctors could input the desired cellular behaviors they want, and an AI would automatically generate the DNA sequences needed, just like how architects use CAD software to design buildings. The implications for personalized medicine are profound. Imagine cancer treatments custom designed for your specific tumor's genetic profile, generated by AI in hours rather than years. The speed of progress in synthetic biology has always been limited by our ability to test designs. That limitation just got obliterated. We're entering an era where designing genetic circuits might become as routine as designing computer circuits, fast, predictable, and scalable. So here's what just happened. Scientists taught AI to read and write in the language of life itself, and it learned faster than anyone expected. The needle in the haystack? The AI just brought a magnet. The question isn't whether this will change medicine and biotechnology, it's how fast. And based on what we've seen, the answer is faster than ever before.